Hi, everyone. Thanks for joining us today. I'm Jen Farmer from the Friedrich's Ataxia Research Alliance, and we're excited to help host a webinar today with Solid Biosciences. And we've got several folks from the Solid team here today who are going to share with you what is part two of a update on and information on the program that they're developing for Friedrich's ataxia. And so we've got Seth Rotberg from Solid, who's going to kick us off, and also Matt Harmlink and June Lee, who are also here to take you through the program and answer questions in the Q&A at the end. So with that, Seth, I think I'm handing it over to you.
Awesome. Sounds good. Thanks so much, Jen. And hi, everyone. Thanks for joining us today for this exciting update about Solid's gene therapy study for people with Friedreich's ataxia called the Falcon study. And just also special thanks to FARA for hosting us today and really for all the, amazing work and support that you guys do for the FA community. If you can go to the next slide, Matt. Awesome. So, for those who don't know me, just a quick background about myself. So, I'm the Senior Manager of Patient Advocacy at Solid Biosciences, and I got involved in healthcare over 18 years ago when my mom was diagnosed with a rare neurological disease called Huntington's disease. And so, witnessing her battle this for 17 years and passing away 10 years ago is really what fueled my passion to help others in the rare disease community and do what I really enjoy doing today, which is connecting with different rare disease communities. And so I'm joined by my colleague Matt. Many of you may remember him from the first webinar. He's Solid Senior Medical Director and Therapeutic Head, Area Head of Neuromuscular. If we go to the next slide. This is just briefly a forward-looking statement. Since we're a publicly traded company, some of this information being shared today may discuss future expectations that may differ from actual outcomes. And if you, after this, just the next slide is just a quick recap of earlier this year. And as Jen mentioned, we presented part one of our two-part webinar series where we introduced our company to the FA community, shared more about who we are, an overview of gene therapy, and our gene therapy approach to FA, including some of our preclinical data. And so you can view this on FARA's YouTube channel by scanning the QR code. I believe the link might get dropped in the chat later as well. Awesome, thank you. And if you go to the next slide, I'll just briefly go through today's topics. So, you know, the agenda for today is we'll kind of discuss again briefly of who Solid Biosciences is, provide a brief refresher on gene therapy and clinical trials. We'll then discuss how our investigational gene therapy is designed to meet the needs of patients and really dive deeper into our Falcon study, including the study design, the criteria, the participant journey, and then, of course, we'll make sure we'll leave time at the end for any questions. You can go to the next slide, please, Matt. So a little bit just about Solid Biosciences, and also one of the reasons why I really, enjoy being here is like we're a life science company focusing on advancing precision genetic medicines for neuromuscular and cardiac diseases. But the company was founded in 2013 by two parents after their son was diagnosed with Duchenne muscular dystrophy at just the age of 2 years old. One of those parents is Solid's Senior Vice President, Patient Advocacy, Annie Ganote, who was also presenting at the first webinar. And ultimately, the company was built with and for patients with a mission to develop meaningful treatments for patients living with Duchenne. But over the last few years, we've expanded our mission to include other rare diseases such as Friedreich's ataxia and other rare cardiac diseases, including CPVT. And so we work closely with patient advocacy organizations and community members around the world to really advance our mission while making sure we include the patient perspective in our everyday work. We're based in Charlestown, Massachusetts, an area of Boston. Currently, our lead clinical program is in Duchenne. It's a phase 1/2, and then we're planning to be in the clinic in FA and CPVT by the end of 2025. And so now the fun begins. I'm going to pass it on over to my colleague Matt, who's going to share a little bit more about gene therapy in our Falcon clinical study.
Thank you, Seth. For those who attended last time, I'm going to do a little bit of a quick catch up for everyone on gene therapy, and then we'll dive into our study. So, if we remember, gene therapy is the discussion is really around how do we treat the cause of genetic diseases. And there's different ways we can do that. In gene replacement therapy, which is where the SG212 program at Salad is looking at it, we're looking at replacing or placing a functional frataxin gene inside of a cell. We know that patients with FA have these GA repeats that's causing the gene not to be able to produce a functional protein. And the goal then is to say, can we add genetic material without changing your genome in a way that then those cells are able to help produce frataxin? Now, I call it that without changing your genome because there is gene editing, which is a different process where you actually try to go in and change the genes themselves that are in your DNA. This is more of a supplement or replacement than that perspective. But when we start talking about replacing genes, the way I like to think about it is first off, the question is how do we get the gene we're looking for into the cell, and how do we get it to the right target cells? So when we talk about gene replacement therapy, we have to first think of what is the delivery mechanism, how do we turn on the gene, and what are we actually delivering into the cell? Now, for our program, and we're looking at a AAV capsid, so it's a adeno-associated viral vector, not necessarily replicating virus. So you're not going to transfect people if you get dosed with this. So, your friends are not going to become zombies with Frataxin. Um, but rather, the idea is that the capsid is there, it's non-replicating, and we know that these viral vectors, there are many of them, all have different selections of what tissues they like to go into, so actually get into the cell. There's preferences there. Based off the selection that we chose, we're looking for a viral vector that we know will affect neuronal tissues as well as cardiac tissues with the hopes of some others, but trying to avoid tissues that we don't want to necessarily transfect or transduce. When you get that viral vector into the cell, though, then you have to say, well, how are we actually going to make that gene start producing protein, right? If you just put it in the cell and does nothing, it has no use. Now, luckily, all of our genes in our body have something called promoters. It's typically right around the gene right before it. And it actually is something that the body can tell to turn on to make that gene become a protein. That's really important because if we think of outside of gene therapy, but just our genes in general, every cell that has DNA, right, has every gene in it. But not every gene we want to express the same in every cell, or sometimes even turned on at all. It's the promoters that allow us to select those. Now, in gene replacement therapy, we can choose promoters then that select different tissue types to express and how much frataxin to express based off that. So it's sort of our second, I would say gateway, but second control into determining how much and where the frataxin is being produced. And then finally, we get to the transgene. So this is a synthetic gene that is synthesized to maximize the production of frataxin as a protein, the full-length protein. So as we think about our program, that's sort of how we're trying to get the goal, getting that frataxin protein in patients with FA to be produced again. And this comes back to this discussion, as I mentioned before, about how do we actually get the gene therapy to where it's going to go and how it works. So the AV capsid that delivers a functional gene will be in your body. It will find the cells and based off different types of proteins on both, can identify these cells in terms of which ones they want to go inside of. And similar to a virus, they're able to get inside the cell and into the nucleus, where they then release this DNA. That DNA, as I mentioned, doesn't incorporate to your genome, but it actually forms typically a circular sort of round structure that then can start to be making frataxin protein because of the promoter that exists. This frataxin protein, then the goal is to then be able to increase the level and hopefully then have downstream positive benefits of what we'd expect in someone who might have more frataxin than those people who have FA. Now, there are differences though, and as we think about the concept of a study in gene therapy and FA, we want to differentiate the concepts of what is a gene therapy versus what is a traditional drug, or some people call them like small molecule drugs. In gene therapy, currently, we think of this as a one-time dose, and there's a couple of rationales for that. One is the idea that if DNA doesn't break down quickly and doesn't change quickly, so once it's in your cell, it should be there for a long time. The other reason is that these viral capsids, when we give them to you because they are similar to viruses, your body recognizes them as foreign. It will develop an antibody to them. And so once you give this therapy, you will develop an antibody to it, and redosing is probably not going to be advised for two reasons. Currently, there's a lot of work going on trying to fix this dilemma, But the idea being, if your body suddenly has antibodies or immune response to a vector, one, you can have more side effects, but it also can block that vector getting in. And that will actually come up later when I talk about antibody testing. This is gene therapy is permanent. It's non-reversible. So it's something that once you take, we can't take it out of your body. It's not something that we can use other medications to get rid of. And so it's a bigger decision of do I want to go down that route of using a gene therapy? Is this the right route for me? The trial timelines are typically about 5 years. Now, I don't want that to be linked to the durability of the therapy. The trial timelines are long, typically longer than the other drug trials, because we really want to follow both from a safety perspective and an efficacy perspective, long-term effects. And we know as we're talking about genetics, these are not always quick changes. These are longer timeline changes. This is something that also the regulatory bodies, FDA, EMA, things like that, they really do want to see that long-term data as well. Now, the consequence of this is, because this is a non-irreversible process, is that it may exclude people from participating in future trials or from taking other gene therapies. If you have antibodies to the vector, obviously that same vector can't be used, but sometimes you can get cross-reactivity or antibodies to other vectors. And because you've had this therapy, if someone is researching the effects of a drug and you've already had this, it may be hard to differentiate, so it may exclude you from some trials. That's going to be a little different than the traditional drugs that we think about, which are the repeat doses. You can wash out of your system, right? You take it in. Typically, it's excreted in either the stool via the liver or via the kidneys and urine. The timeline of those trials can be shorter sometimes. It depends on how long you think the effect of the drug will take to see. But there's oftentimes a washout period afterwards in those drugs where you can say, once the drug and the effects are out of my system, potentially I can go into another research trial. Now, I do want to call it in this discussion, this is focused on research, not on clinical treatments. So the piece about the permanence and one-time dosing, that is certainly very much clinically relevant, but how people in the clinical world use combination therapies may differ than what we're talking about in the research world as well. And that's because how we approach patient care is different than how we might approach research. So for those questions, certainly feel free to chat with your physician and your FA specialist. So with that concept in mind of what are the differences of gene therapy as compared to what we might think of other drugs and timelines and things like that, the question that I would think of is, well, what's the first step in how we come to the point of saying we want to actually do a study in humans? Before that, oftentimes, right, when you start off as a clinical development, we think of what is a good target, what is a good gene, disease, do we have a mechanism, things like that, conceptually. And then that moves into a field of what we think of as preclinical. Some people call this non-clinical. For the SGT-212, this has been evaluated in preclinical studies of FA. And preclinical studies are important. They determine potentially route of administration, safety, potential efficacy, both on timelines and desired or expected effects. In our preclinical studies, we studied mice and non-human primates. People think of that as monkeys, to confirm that SG-212 may support frataxin expression in target tissues, increase mitochondrial function, improve cardiac and neurologic function, and increase survival. So from these multiple studies that we did, the idea was that saying we are seeing signals that are very positive and reassuring. And so the next step then would be saying moving forward is to get clearance from a regulatory body, which we announced back in spring and late winter of 2025, saying we can move into humans to do a similar research trial. And that brings us to the Falcon study. So the Falcon study is a study to evaluate SG-212. That's the name we're using for the treatment, the medication. And the next couple of slides, I'm going to go through what is our study overview, who can participate, your journey, what will be required of participants. And then I will talk at the end about this dual route administration. And I'm going to save that to the end just so we can really hone in on some of the pieces of what does it mean to be dual route versus just a single route of administration. So to start us off, what is the study overview? Falcon is a phase 1b open label multi-center trial focused on evaluating safety and tolerability in SGT-212 in patients with FA. Phase 1b studies are early studies looking at smaller patient populations where we really want to test, as mentioned here, the safety and tolerability as we move into human trials. It's open label, meaning that all participants will be getting drug. There's not a placebo arm, things like that. And the multi-center, just saying there are multi-sites that will be doing this trial with us. The study will begin with non-ambulatory participants, followed by ambulatory participants. We're doing these in cohorts, so there'll be a few patients that we're dosing, first who are non-ambulatory, following the safety and tolerability initially, and then moving beyond that to ambulatory participants there. It will be delivered via this dual route, one which will be an intradentate nuclear infusion. Now, this is an approach where we can actually directly infuse the tissues. The dentate nucleus is the sort of the back of the brain. You have your cerebellum, which controls coordination, and in the middle, there's a couple of nuclei, and the dentate nucleus are very involved with the Friedreich's ataxia disease process. And so the goal is to actually directly infuse into those dentate nucleus, as well as an IV administration, so through a vein. And I'll talk a little bit later about why that is and why physiologically that might be beneficial. All participants, as I mentioned, will receive the study drug. And from pre-screening to study completion, participants will be followed for around 5 years. The next question is, who may participate? So in this initial study of the drug, we are looking at either male or females, age 18 to 40 at screening, either ambulatory or non-ambulatory, though we do have an order in the study that we're following in evaluation. You cannot have received gene therapy at any time because we want to make sure that we're really studying our drug in, as it is. You must be negative for AAV antibodies. And I'll mention that in a little bit about what that means and how that will be tested. You obviously have to be diagnosed with FA and have had symptoms start at age 25 years or younger. There are some objective functional and cardiac criteria as well. That as you get involved in this, people who are interested in the study, as you talk to the study sites, they can explain that more and how they'll evaluate for those pieces. Now, importantly for Skylaris or OMAV, we are allowing OMAV in the study. However, we do want either patients coming into the study to have been on a stable dose for at least 12 weeks and plan on maintaining that dose throughout the study, or if they're not on it or want to stop it, have done that at least 12 weeks before. And that again is really over this initial dosing period, which you'll see in a little bit has a lot of monitoring and evaluations. We want to make sure that we're doing the best and safest for our participants in terms of being able to follow safety signals and things like that. Additionally, throughout the course of the study, especially the initial 18 months, you cannot be on other investigational or approved disease-modifying FA therapies. Now, this is not Tylenol or typical heart medications. This would be medications that would be specifically for Friedreich's ataxia. If they're approved medications, things that actually change for frataxin or things other like that, exceptions being Skylaris. So that's the initial criteria as we start. And I think I'm going to then move us into what is the participant journey looking like. This is a lot to take in at once. We'll walk through it sort of step by step as we go. And if there's questions at the end, happy to jump back to this to talk more. So as we start off with for a participant, we have our pre-screening. So before someone even is saying they're eligible for a trial or getting into dosing, the first question we would be looking at is, if interested, and every site handles this will handle this a little bit differently, is being able to do what we call pre-screening. And this is essentially sort of a small subset of screening tests, in this case, looking at the AAV antibody testing before someone really is committing to screening days, which tend to be in trials and in ours certainly is much more involved, a lot of functional tests, other blood tests, things like that. The pre-screening has to be done within 45 days before dosing. And that's because AAV antibodies in the general population have been shown in people not dosed with gene therapy in adults to be about 30 to 40% of us will have positive antibodies from other reasons. And it's not always because we've been exposed to an AAV, but just because there's cross-reactivity and other rationale for that. We know then that there's a thought of, could someone convert over, be negative and go to positive? And the longer you'd wait between those tests, in theory, the higher likelihood this would happen. In the data that we have, that is very low rate in the pot, and I'm talking about not solid data, but in general, the field, the risk is very low, but it does exist. And so for that reason, we want to make sure we're being very conservative with that 45-day. If you would test in the study pre-screening and be negative, and the dosing day would be after 45 days, you just have to repeat the blood test. The risk is very low, and that would be done at screening if we think it's going to be later. But this will allow people who are interested but really don't want to have to commit to, oh my gosh, a full screening day and all that activity, if this might be something that would screen you out and not make you eligible off the bat. After that, you'd move into our screening period, and this is prior to dosing. And in this case, there's sort of two parts of the screening, is the way I think of it. There is the testing, which is saying, are you eligible to be dosed with this therapy? Is it safe for you to be dosed? And do you meet the criteria that we're looking for the right participant for the study for both safety and for the potential other tolerability and other signals that we're looking at as well? That will include some blood tests, some physical exam features. We'll be looking at some cardiac features, as I mentioned before, and neurologic functioning. And because we're doing ID and dosing, also imaging of your brain to look at the structures there. Once those assessments are complete, and if you qualify for the study, you would then move into the next part, which is dosing day. So on day one, this is where someone would be dosed. They would be admitted to the hospital for the neurosurgical procedure, and you get the IDN procedure, so the surgical infusion, followed by the IV infusion shortly after that. Patients will be staying in the hospital for a short period for very close monitoring over the first initial days before being discharged home. Now, post that discharge period, around day 6 up to day 60, we have fairly frequent outpatient visits. This is a period right after dosing when they're in gene therapy in general. You can see signs of inflammation. People may not feel as well. And we want to be very cautious and be cognizant of making sure that we're following our participants who are getting dosed very, very closely. This would mean things like a physical exam, talking to the study doctor and having frequent visits, as well as lab values and having some blood draws to see if there's signals that maybe you aren't having clinical symptoms of, but might be indicative of we need to adjust treatments, things like that. As we move towards day 60, we get beyond that expected period, and then we get to sort of start spacing out visits a little bit further as we start looking beyond just that initial safety to other factors. And this goes in then to that day 60 all the way towards year 2. Here we have a visit on day 90 for assessments, and I'll mention some of those in a minute. We have a month 12 visit. This is our primary safety outcome for the study. And so this would be really looking at is the drug safe and tolerable in the first year, though we obviously continue to follow after that as well. At month 18, we have another strong point for other assessments that we want to be able to follow. And then as we get into year 2 to year 5, we start spacing these out to be every 6 months, where these then become sort of a medication review, some lab testing, a physical exam, some functional assessments. And then once per year after that, we also do a heart and brain imaging and assessments as well to follow the longer term effects of the medication. There are biopsies involved in the study, specifically a cardiac biopsy, and I'll mention those in a little bit about what that means. Okay. Now, what does it mean beyond the biopsies and the accommodations and things like that? And all the different study protocol pieces, what does it mean to participate? What's the time commitment that we're asking someone to do? It is a longer study. It's a 5-year study. I absolutely understand that. So it is a big commitment, as is gene therapy. The dosing, as I mentioned, is multi-day in the hospital. The visits that occur after the procedure, there's about 20 visits over the course of 5 years, with more visits initially, and then they start getting spaced out as you go further. Um, Now, we are cognizant that these type of studies can be burdensome on families and participants and your caregivers. And so travel expenses from your home to the study site are things that we want to be able to support, as well as accommodations and food during your stay. And our goal is that you and your caregiver should not have to pay for study-related expenses as we go forward in the trial. Because if we then take the large view of the trial and bring it down to what to expect during a single visit, a single day, right? You have done your pre-screening, which I mentioned is more of a blood draw, and then you come in for either screening or those assessment visits. What does that look like? Well, because this study has a goal of looking at safety as treatment, we want to look at that, but we also want to collect information not just from an exam and blood draws, but also how you feel you're functioning. Your well-being is the most important. And so one of the things that we also understand, and having spoken with different people with FA in the community, is fatigue is an issue, and fatigue can be variable. And so you don't have to complete all these tests on one day. We do have windows assigned for the study so that we have a little bit of flexibility. Obviously, we don't want to get too far out of two tests that should be linked too far apart because they may not scientifically be able to be linked. But we want to have enough flexibility that you're able to get these evaluations done in a reasonable, appropriate manner without wearing you out. So what are we going to be doing? Well, beyond just meeting with a study physician, doing an exam, chatting with them about how you're feeling, there are going to be some blood draws. These look for changes to your immune system as well as other molecular markers, biomarkers for FA, things like that. In addition, we want to look at that heart and brain function. We're doing dual route therapy with the goal of affecting multiple organs in our body. And so because of that, we really want to be able to say, are we be monitoring these very, very closely. So this will be done with MRI. We want to see how the treatment might impact the function and structure of your heart and brain because of that. And they'll be done periodically throughout the trial. In addition, we will have some functional tests and questionnaires. These will both be able to show us how you're performing, but also tell us how you feel you are doing with the medication. And I think both are very, very important as we look at the whole picture of how SG212 may be effective in FA. And then finally, we do have some biopsies. I will call it, for example, we're doing cardiac biopsies. And this is because if we're giving this gene therapy and we are trying to get protein expression, hopefully with the goal of treating symptoms, we want to be able to actually look at the cellular level to see what are we seeing. Can we see those types of markers as well? Now, for those who aren't familiar with cardiac biopsies or even that ID in neurosurgery, these are periods where you would get anesthesia or sedation in discussion with the doctor who's doing the procedure, yourself, your FA specialist, in terms of what's best for you. Cardiac biopsies are, and I'm not a cardiologist, so please excuse my poor description of the biopsy, but the cardiologists who do this typically take a small wire. They actually can go through a vein, typically in your groin, up into the heart, and it's almost like an alligator pincer. They can take out tiny pieces that are about the size of a grain of rice. They take a couple of those at different points throughout the study as we go forward. So very small pieces relative to what the size of our heart is as we go forward. But I think that's important then to touch on because we are doing these two different routes. What does this mean? It is a dual route therapy. One route that we're talking about is our intradentate nuclear infusions. And so as I mentioned, the dentate are these small little structures in the back of our cerebellum. You can sort of see a picture of the cerebellum there. For those who aren't familiar with cerebellar function, it helps with control of our movements and a lot of the ataxia that we see in FA and with other diseases coming from that overall structure, with the dentate being particularly affected in FA. So that's where the surgeons can go in directly. Also with this IV infusion, we can then systemically dose. Now, one question may be, why would we do that? Well, one interesting thing about the human body is we have the blood-brain barrier. So around our brain and our spinal cord, we have this very strong tissue barrier that prevents or reduces the amount of things that can go between them. As a neurologist, it's great that we have this because that way, every time we get a viral cough, we don't get meningitis. However, it also means that as we try to get medications and treatments from the systemic system, the blood, for example, into the neurologic system, we have this extra barrier we have to overcome. And there's different ways you can do this. Sometimes you can use drugs that you can dose, or you have to go higher enough to sort of push them across. In our study, we felt that after doing a lot of the non-clinical work that I mentioned before, that actually doing direct infusions would be the best way to be able to get the drug to where we want it to be. And so that's why we went that route. So what is this intradentate nuclear infusion look like? So what would happen with a participant is they would come in, they have an MRI beforehand of their brain. That MRI actually can be moved over with the neurosurgeon, and they can actually look at all the different structures and map and model where they want to go. When someone would come in for their infusion that day, they'd be under anesthesia, they'd be asleep, but the surgeon then can actually map out the shape of your head and overlay the two. By using that, they can use this robotically assisted device that helps them look at what is the exact trajectory and depth they want to be able to put the catheter into your brain to infuse. These are very, very small catheters as well. That robot then helps assist. You can sort of see where it has on the leftmost slide, the robot and the array. In there, they can make a incision where they put a little bolt. It's not a bolt like in a chair, but there's actually a hollow tube in the middle of this bolt into the back of the skull. It allows insurance that the catheter that they're going to put through with the drug goes through exactly where they want it to be. From there, once that's secured, as you can see in the middle slide, they will then take a small catheter where they can actually place it down to the exact depth they're looking for. Again, they've mapped this on the MRI, so they know where the dentate nucleus is. At that point, this can be confirmed by imaging. So they can actually look and say, yep, the catheter tip is where we want it. The infusion occurs. During the infusion, if they need to also do imaging to make sure we're infusing the right area, they can. And certainly afterward, they can use imaging confirmation as well to look at the drug and where it's at. Because the drug is actually mixed with a contrasting agent, just like if you got another MRI or a CT scan at some point, there's many contrast agents, there's specific ones for these type of procedures. At that point, then we can actually see that on the MRI where the drug is relative to other structures and the dentate nucleus in your brain, and be able to see that it's in the place we want it to go. So it's a very precise MRI guided infusion to be able to get the drug to where we want it to be. And I think that precision is something that's important for us in making sure we know exactly where and what we're infusing with SG212. Now, after the procedure is over, they take everything out, the bolts come out. You would get either a little suture or maybe even a steri strip on the top, and then you'd wake up. Because of the protocol, you'd stay in the hospital for a couple days subsequently. This is obviously much more complex than our IV infusions, which many people are probably more familiar with how to do an IV infusion. You have an IV placed in your vein, a small catheter. At that point, the medication would be infused in there. One thing to note, it is the same medication, but the doses are not the same because we're able to titrate and modify based off what we think is the best dose for the systemic dosing versus the best dose directly into the dentate nucleus as well. So we are able to separate those as we go forward. And the systemic treatment has the goal to deliver therapy to the heart to address cardiomyopathy, which we know is a leading cause of mortality in FA. So to summarize that for us today, the study duration, as I mentioned a couple times, has been about 5 years. The study drug dosing, we do this 2 doses within one in-hospital stay for the study, with about 20 visits over that 5 years, the majority in the first year. Study drug administration is done in those 2 different routes that we're talking about. And then one thing also to note is that because whenever we give a viral vector, there is a chance that you'll have an immune response. Your body will realize it's foreign and fight it. You do go on steroids just around before dosing to a little bit afterwards. It's not for the full duration of the study, for a shorter period of time. Um, and to help suppress that immune system and avoid symptoms or signs of that immune response. We are having sites in the US only. And one question that may come up is, do you accept international patients? It's site by site specific. As a study we do, but each site, that'd be something you'd want to reach out to them as we go forward about. Um, If there's other questions of information, please feel free to email the info@curefa.org for now. We are, in terms of timeline, and that's another question of when is the study going to start up? What is our process there? As Seth mentioned, our goal is to dose our first participant by the end of the year. And most of you looking at the calendar know we're getting pretty close to the end of the year. We have not disclosed the sites yet because we want to make sure they're fully up and ready to go. That should be very shortly, but we want to respect they will tell us when they're ready for us to give their names. We also want to make sure that if you reach out to them, you are getting the best response in terms of they're ready to be starting to talk about accepting participants in the study. And with that, I will end things right here. Thank you so much for letting me chat. I'm sorry if I go very fast, but hopefully there's lots of great questions afterwards that we can talk about.
Matt, thank you so much. You did a really nice job taking us through a lot of information, and I know there are some questions that are coming in, but maybe while those questions are coming in and Kellen's queuing them up for us, I will maybe just reiterate kind of the next steps in terms of if people are interested. FARA is willing to kind of generate a list of folks who are interested with your email addresses. And then as sites are able to open, as information gets approved and put on clinicaltrials.gov, and we have clinical coordinators that we can refer you to, that's what we'll do. We'll have the list of people, and then as sites open, we'll email you to make sure you know that they're open. We will also be posting on our website on the clinical trial finder this Falcon study, and we'll announce the sites on the website. We'll put it in our monthly newsletter. So there will be multiple ways for you to get information about the study. But if you want to make sure you get it right away, I think the email is probably the best bet for now. But we will continue to share information with the community. So I'm also gonna, Matt, there were 2 questions I had based on things that you discussed. So one thing you mentioned in the inclusion exclusion criteria was the AAV antibody screening. And I think you said there was a chance like 30 to 40% that people have already been exposed and might already have antibodies.
Mm.
Is there a way for people to know that ahead of time? Like, could you go to your doctor and just ask, like, can I get tested?
That is a great question. There are some commercial antibody tests, but I will say you have to make sure you know what antibody test you're testing for. Not all the AAV antibody tests are currently commercially available. Um, it is something that I think if someone is interested when they start communicating with the site, I say this because I want to make sure that we're testing the right antibody. You don't just go to your PCP and say, "Can you test me for antibodies?" Because they might test you for something else. Not that I've seen that ever happen in my career before. But I want to make sure that is a component. But yes, people can be tested for different antibodies commercially. The other difficulty, to be very honest, I don't know what insurances will cover because that wouldn't be a trial related activity. So it may be more pricey if it's done that way, versus being done via the pre-screening route.
And so that my second question was about the pre-screening route. And does somebody have to travel to a study site to do that pre-screening to get the antibody testing, or is that something that can be done at home?
So that is a site-by-site specific question, unfortunately. And it depends not only on the ability of the lab, but will sites, it's maybe more nuanced than people want, or maybe they want to know. Some sites are allowing remote pre-consenting and allow that. Not all sites, institutions, academic institutions allow that. They want you to actually come to be consented in person. So as we have sites opening up for enrollment, that will be a question people will want to ask as well. But I think that's a great question. It is feasible, as of maybe what I'll say, to have some of these things done so people don't have to travel.
Awesome. Well, thank you. And I think there were one or two questions I saw early on that were related to AAV and the antibodies. One of which was, if you participate in the trial and you get the gene therapy with this vector, does that prevent any future use of vaccines that might use the AAV virus for the vaccine to be delivered?
Um, so I think so. I'm going back to my general pediatric days. Most of our vaccinations aren't delivered by viral vector. They may be, again, they're especially AAV vector. So I think I wouldn't anticipate that a vaccination would be at risk. However, getting dosed with a viral vector will likely make you non-eligible for future doses of that same vector because of antibodies. And in the field of vector dosing, there's more literature coming out about cross-reactivity with other vectors as well. So if you get dosed with, for example, AAVX, AAVY, and X and Y are made up. We use numbers instead. You may also have antibodies to that. So it is something in that case. That's why it's a big decision for people to make. I don't want to downplay that at all of what this means going forward.
Yeah. Thank you. All right, let's see. There were a few questions, I think, about some of the other inclusion, exclusion criteria around, I think I just saw one around age. And let's see, Kellen, I'm going to bring you in. I know you've been looking at these more closely.
Sure. Um, I did see another one related to the viruses actually, if we want to keep with that theme. So you just said that we aren't sure about like if gene therapies are delivered with different types of AAVs, then that might make it so you can't have two of those. But what about in the future if there's a gene therapy that's delivered by a virus that is not an AAV? Do we know anything about that?
I don't think anyone has studied it. This is you're sort of asking me of things outside the solid realm, but as maybe scientifically, I think there's not a lot of literature there. It's the same idea of the two viral vectors. While we assume antibody positivity would occur, there's also a lot of interest about how would we treat patients either redosing or dosing with another viral vector. That's been going on. Salad has presented at a scientific conference about some of our internal work we've been doing as well on the same topic, not necessarily even with FA, but just in general in the field. So yeah, I hope that answers the question.
Thank you. So we did have a question about the age inclusion criteria. Could you go over that again?
Sure. And actually, if I don't make everyone sick, I'm going to try to go back to that slide real quick. So you can sort of see it here. So currently in the study, our age criteria is you have to be between 18 and 40 at screening. Again, being a longer study, we understand that people may become over 40 throughout the study, and having had symptoms start before the age of 25. That's your symptom start. That is not your first neurologic visit. We totally understand that people sometimes will have symptoms for a longer period of time beforehand. The reason for this, and I'll both talk. I think I saw a question on the older, over 40, and I'm assuming people also ask about younger, under 18. As we start moving into this is more of the drug development piece, as we start moving into a therapy with using a clinical trial in humans, we want to be very thoughtful in terms of how we're starting this. Does not mean that's the only ages we'd ever consider. But initially, we're being very thoughtful with a small study as we look at safety, tolerability, how does the study look, how does the drug work with patients with FA, before we then say what is the broader scope that we want to be discussing with the community and with the science, both patients as well as scientifically.
Thank you. And we have a kind of related question, but can you explain the rationale behind beginning with non-ambulatory patients?
So yes, I think, and I guess maybe I'll start it off with, this was actually at the advice of the FDA in discussions with them. They really wanted us to start with non-ambulatory participants. Part of the rationale was, as we look at a safety study in particular, they wanted us to look at patients who maybe are further progressed in the disease state, as we start to dose before we move on to ambulatory participants. It's a risk benefit potential. If there's a slight change in ambulation gait, someone who's not ambulatory may have a different profile. We do know we want to look at both groups as well of participants. We don't want to just stay with ambulatory participants or non-ambulatory participants. Excuse me. But we had to sort of also choose which group we're starting with first, because I think there is value in looking at the whole course of disease. I mentioned ages as well, but also functional status. That has been used in the field quite a bit as a demarcation of changes. So that's something else. And because of that, following that advice, we sort of opted to start with our non-ambulatory participants first. Again, not that we're not going to go into ambulatory participants. We opted to just also do this as a cohort so that we can be very thoughtful as we look at groups of functional statuses together.
Yeah,
And
and I
do
think that gives
you-
us, part of what we try and learn from these early phase studies is what to look for in later studies where we have to prove efficacy, and what we might see in efficacy could be different, right, in people at different stages of the disease. And so that's the other reason, I think, to kind of cast a broad net early on, to know what potential benefit could look like. There was another question in terms of how many people will be enrolled.
So currently, the entire study is looking at about 10 participants between all the different cohorts that we talked about. I will put an asterisk again on that, that is for our phase 1b initial study. As we go forward, as we get more data, that we'll certainly have discussions about the overall program and what that means.
Jen, did you see any other questions related to inclusion criteria? Should we go to another topic?
See, maybe just one question that I saw in here that I think is a quick clarification. Somebody was asking if it was essentially the same drug being administered with the 2 routes of administration, and it is the same drug, correct?
Yes, it is the same capsid, promoter, and transgene, so the same drug versus in both routes.
Yep. Yeah, we can go to,
We had a couple, we actually have a lot of questions about the dose of the drug, and I think some people asking questions are either scientists or science enthusiasts because they are great questions. One of them is just simply, is the dose that every person receives in the trial going to be the same, or is this the type of study where you're looking at different doses across the participants?
So I don't think we publicly disclosed the dose yet. So in the dose ranges yet. In a cohort, everyone would be getting the same dose. I can be fairly confident to say that, as we want to study it across the participants. As we look at taking a step back to a phase 1b study, that is something that we will want to follow all the data, both safety and other efficacy signaling, to determine what is the best and safest dose to be dosed. So in the course of a study, it may not be always the same dose. And I'm not saying that our study has already had that included, but more in the general concept as people are thinking of the study. I'd encourage anybody who is a participant to chat, if they're interested, to chat with that PI who will be able to share more information about what their thoughts are there.
And you said you can't disclose what the dose is, but are you able to explain like how a dose is decided? Does it have to do with someone's weight? Does it have to do with the size of their dentate nucleus?
Sure. So maybe to take us, I think the question is getting at sort of how do we vary doses by participant then potentially. Um, so dosing in general in gene therapy, so taking a step back from just the program, because I think that I can speak more broadly and this will help, is you want to dose based off what your amount of target tissue you're looking for. So for example, in a systemic dosing, because we know there are larger people and smaller people in the world, most people want to shift according to weight in terms of how we look at dosing. That's very standard across the field. As we look at brain target tissue dosing, it's a little different because in that case, the dentate nucleus is in adult brains. We go back to, for those who love anatomy, our brain is fairly same size. We just stop growing the size of our brain when we're still a younger kid. And so by the time we get to adult ages, our head size doesn't change its shape and size. However, our dentate can vary a little bit in terms of shape and size. And so there's some more um, complex discussions around how do you get great coverage and things like that. That's why the neurosurgical approach is so precise and why we want to do that rather than doing a more broad, give a lot of drug, hope it gets where it goes in the brain type approach, I think is what our goal is. So that is some of the discussion. I think as a clinician scientist, that's something that's very interesting to me as we look at overall at these type of programs and dosing in terms of how do we find the best dose for participants. So I know I didn't answer the question specifically, but hopefully giving whoever asked it enough background to sort of understand how we're thinking of dosing as we go forward.
Thank you.
I see a question kind of coming a little bit more about like what happens after. And so like, do you need to, is there any special restrictions, or the question is, what care do you need after the infusion? Can you be active as you were beforehand?
Those are great questions. And some of this will be dependent on the surgeon. I will defer to them because they do the procedures. I'm not a neurosurgeon, and I want each individual participant to have that discussion. However, in our discussions with the neurosurgeons, with these type of procedures outside the trial, most people recover very quickly. Most of the time, they go back to activity fairly quickly within days, is what we've heard, and to have that discussion, which I think is important about the approach. In terms of restrictions, the one thing in the field of viral vector therapy is that anytime you take something in like this, you can shed it. That's a common understanding of we shed it in different saliva, urine, stool, things like that. As I mentioned before, these don't replicate, so you're not going to infect your caregiver and they're going to start having this. But if in theory they are exposed to it, they could develop an antibody against it. Now, this is something that people have looked at scientifically. The expected risk is incredibly low. Every site has a different view of how to manage that. And some physicians are more conservative, some are a little bit more okay with doing other activities. And so that's a discussion with your site of what do you want to do? What does it mean? What's the risk? it also is a discussion with what your caregiver gives care for, to be quite honest with you. And so, but yes, there are some of those pieces. There may be some modifications post-dosing that way as well to discuss your activity level.
Is there a period of time that is well established, at least for, you know, in terms of exposing other folks to any, you know, viral shedding, like things like that, that after a certain number of days, that doesn't matter anymore? So I'm just thinking about folks who might have siblings with FA
Mhm.
and things like
Yeah.
that.
Yep. That's a good question. It varies a little bit, I think, by the capsid. And that's one of the pieces of any study is also looking at that in humans, how we shed. In the field, typically we think of the highest risk is within the first couple of days, especially if you're looking at saliva and mostly. Stool can be a little bit longer. Not relevant for this study, but in gene therapies in babies, that becomes a bigger topic when you have to change diapers. Um,
Uh,
so something to think about in that case as well. That can sometimes last for a couple weeks, but it does exponentially drop in the studies that have come out. And so again, the risk also drops significantly. Most institutions will just ask that for those type of things you wear gloves if you're working with
yeah.
secretion, things like that is enough.
Okay. Um, there were a few other questions, kind of related to timing, you know,
Mhm,
because these are such long studies lasting
mhm.
5 years. I saw a few people ask about, you know, do you have to wait the full 5 years before you could start another study, like an efficacy, a phase 2, 3 study, or could those start sooner while you're still in that 5 year time frame of monitoring outcomes from this study?
I love these questions because they're actually clinical development questions, which are great. The answer is I can't predict what the company's going to do, right? There's a lot of variables there. Again, in general, you don't need to wait for the duration of a 5 year study before you look at future studies, evaluations, expand to other patient groups. Those are all things that are a constant discussion of when we think we have enough data, when the regulatory agencies feel we have enough data, that we can start to look at, the whole population that we were really interested in. So short answer is we don't have to wait, but it will depend on a lot on what we see as we go forward.
That's good. I just knowing some of the folks who gave those questions, it's a matter of urgency, right? We want these
Mhm.
trials to move forward faster and thinking like, oh my gosh, we'd have to wait 5 years.
I absolutely understand. And I'm sorry if I ever hedge as a neurologist, right? It's sort of been trained in me. You always hedge a little bit in everything, even if it's, my kids telling me they have a headache. Um, but so if I do that, I apologize. I also want to be very transparent with the community of as I approach this study, this program, to be participant, every data point is something we want to watch and follow and evaluate and analyze to make sure that we are gathering all the data, using it, and it's really helping us drive forward with what is the best route to go.
Helen, are there other groups of questions that you've seen that you want to ask
There
next?
there are. So I think you said at the beginning of the presentation that as a phase 1 study, the main goal is to understand safety and tolerability.
Mhm.
But we do have a couple questions about what are potential effects of the gene therapy, recognizing that that isn't the main goal of the study, but based on maybe the preclinical data or other data that you have, can you talk about potential effects? Can you talk about what other organs it might reach? I think someone mentioned the dorsal root ganglia specifically, if it's going to get there.
Those are great questions. So we have a lot of data, unfortunately, and some of it has not been made public. And it being a publicly traded company, as Seth sort of alluded to in the beginning, I'm restricted on some things that I can say. Um, we do have a lot of data that we've studied both in this program and in other FA programs, looking really at what is the best route to go. Um, the To answer the question of sort of other target organ tissues, and I think maybe more so of sort of this looking at safety, whenever I think about moving from a non-clinical world, mice, non-human primates, monkeys to humans, there's always a difference, both in the animals themselves, but also in what we think of the disease, right? In FA mice, they behave a little differently than FA would in humans. And so the question of, I think, efficacy, though we may have seen some of those signals in animals, how we translate them clinically, right? How I would have told my patients in the same scope. It's hard, I think, to make that assumption now with no human data. And I know that's what someone who might be thinking about the study really wants to dive into, right? You want to know, what could I see in terms of the dorsal ganglia and possibly some of that sensory neuropathy that comes with it? Is it going to affect the spasticity of the spinal cord, the dorsal, columns, or from the fatigue even in muscle? I think that's hard for me to make assumptions and jumps to, and not that I don't want to, but especially because I do have to be cautious as a employee of a publicly traded company for now. That is something that I think, as I mentioned, we chose doses, and obviously not just for safety, but also for the potential for efficacy. But as we move forward into the study, that is where we'll have to be very cautious and watch. Obviously, we want to see some of those signals. We'll be evaluating for them as well. But I don't want to, I'm, again, as I mentioned before, I hope I'm not being a negative Nelly on the talk. But I want to be very open with anybody who's interested, especially those first couple participants, and how brave they would be to be coming into a trial with the data that we have, right? And be very transparent of what data do we have, what what can we predict and expect going forward. So in terms of that, we do know that, again, that being said, the other piece I say that with that is that because as we look at FA as a clinical disease, it is very complex, and one target organ like the dorsal ganglia as an effect in ataxia, I think there's multiple parts of each of these systems affecting someone's gait, for example, and not just, we can't just call it one organ or another in terms of that piece from a medical perspective. And so that's also why I'm hesitant to say, oh, DRG there might be affected, so that might help this symptom. I don't know, and I will defer to the experts on the call and the experts I've talked to elsewhere. But that's just what I believe with FA we'd probably be seeing. And so differentiating is a little hard by organ system versus sort of saying in general. Sorry if I ranted a little bit about that, but I do, sorry, I just want to make sure I'm very overt with anybody interested. And those are discussions, if you are interested, I would encourage, as you talk to the PIs, right, ask them those great questions as well. As you get more engaged with the study before dosing, those are things that you will have discussions with in much more detail through the screening process as well.
And so it sounds like there's some data from the animal studies. This study will look at potential efficacy signals, but the plan would be is if this study goes well, there would be a future study that would really be looking at effect.
Currently, yes, our primary endpoint right now is safety. We want to obviously gather as much data from anybody who's willing to participate, whatever they're willing to share with us. But as we go forward, as we look at larger data, and that is all going to be part of that program development as we get that first couple participants and we grow the program of evaluating, discussing with sites and participants and experts, and making sure we're also using the right endpoints and the right biomarkers, and all those pieces are really key.
Thank you. We only have a minute left, Jen. I don't know if you have any last questions you want to end with.
Maybe not a question, but maybe just kind of reiterating for folks. And first, let me just thank Matt and Seth and June for being here today and sharing this program with us, and for all the work that you and your colleagues have done on this program. I know there's been a lot of work behind the scenes to get us to the point of this clinical trial. And, this is something that is really new for the community. And Matt, really appreciate your being honest about that and encouraging people to ask a lot of questions, meet with the study team, do more to understand what's involved in the study. But just to reiterate, this approach is, this dual route of administration is really to try and, allow us to make sure that we can deliver a gene replacement strategy to an area of the brain that we know is affected in FA, and at the same time also treat the other organs, because you only get that one-time approach, right? So there's a lot that's gone into sort of thinking about the risk benefit trade-offs when it comes to gene therapies. And, by having this direct route of administration, you get to smaller doses, you get to precision, and there are different trade-offs that went into this. And I think people are realizing that just based on their questions, but just want to thank you and the team for all the work that you've done to bring this program forward. And as we mentioned, people can email us at info@curefa.org and ask to be put on the update mail list, and we'll send you updates as we get them, as sites open. So you can reach out to the site investigators for more information as well. And just thank everyone. There's really a lot of interest in the study, clearly through the Q&A.
Thank you, Kellen and Jen, for hosting us. This was great. And I do appreciate the questions. They were great. And I appreciate the engagement. And please, as we continue to have these discussions with the community, bring more. They're wonderful to have these chats.
All right. Take care, everyone. Have a good afternoon.
Thank you, everyone.